Is we're going to talk about the genetic regulation and how transcription, translation, and gene expression becomes regulated. The next section is talking about genetic regulation. So we're going to discuss the processes by which the expression of genetic information, i.e. transcription and translation, is regulated. I think you can reason that if these processes become malfunctional, that you're going to have major problems inside the cell. If, for example, the genetic regulation becomes as such so that proteins are made too much, it could perhaps lead to cancer. Whereas in another situation, if not enough proteins being made, that could cause to a dysfunction or a deficiency in some sort of protein factor. Let's begin our discussion with the lac operon. Historically, the lac operon is the major illustrator of gene expression. Now keep in mind, however, that the lac operon is specific to prokaryotes. This isn't a situation that happens in our cells. It's a situation that happens in particular in E. coli. It's a system that's designed to operate when E. coli runs out of glucose as a carbon source. So let's look further. In the lac operon, the major sugar that bacteria prefer is, of course, glucose. Okay, glucose. If bacteria run out of glucose as a carbon source, then they will begin to utilize lactose, illustrated here, as a carbon source. But in order to do so, the bacteria are going to need to turn on a set of genes that are involved in metabolizing lactose. Okay, the cells love to conserve energy. Nobody likes to waste energy. So the E. coli, the bacteria, in the absence of lactose, there's no need to express the genes that metabolize lactose. They have everything they need for glucose. So in order to metabolize lactose, there's a series of things that have to happen. Number one, the bacteria have to take the lactose and bring it into the cell. That's the job of an enzyme called a permease. It helps bring the lactose into the cell. The second thing the bacteria have to do is to take the disaccharide lactose. Remember, lactose is a disaccharide consisting of galactose and glucose linked together through an O-glycosidic linkage. They have to be able to break the lactose into galactose and glucose, these monosaccharides. That is the job of an enzyme called beta-galactosidase. It's also known as lactase. But in bacteria, we call it beta-galactosidase. So once the galactose and the glucose monosaccharides are created, they can both be shunted into a pathway of glycolysis so that the bacteria can utilize the sugars, oxidize the sugars, generate ATP energy. So in a typical happy bacteria, there's no need for this. But let's say that lactose is completely unavailable. It's completely unavailable. How is this pathway going to be shut off? Okay, this pathway is going to be regulated in the following manner. There's going to be a set of genes here. If you remember that prokaryotic messenger RNA is polycistronic. So there's a set of genes, we'll call these genes Z, Y, and A. These genes are going to be what we call coordinately regulated, coordinately expressed, so that when you get transcription off of this promoter right here, when you get transcription, you're going to get this messenger RNA made. This messenger RNA is going to encode 3 distinct proteins, one of which is a beta-galactosidase, the other of which is a permease, the third of which is a transacetylase. Don't worry about its function. Just know that transacetylase is an important protein in lactose metabolism inside bacteria. So, in a typical situation, by the way, these 3 proteins then are going to be made from one messenger RNA transcript, i.e. polycistronic messages. So, in a typical situation, the bacteria want to keep very low. They want to keep the expression very low for genes that are metabolizing lactose. How do they do this? There's an important part of the promoter called the operator region. So you have a promoter, and then within that promoter sequence, there's what's called an operator region. It's a specific region of that promoter that is important for regulation. When lactose is absent, there's going to be a protein made called a repressor protein. So the repressor protein is going to be active. The lac repressor protein, it's being transcribed and translated from this I gene. This lac repressor protein is going to be transcribed and translated. It's going to be active, and it's going to bind to the operator sequence. The operator sequence then is then going to be bound by the lac repressor protein. The lac repressor protein, I want you to think of it like a roadblock. It's a major roadblock that's simply impeding the RNA polymerase from coming in and from making messenger RNA. Okay, the lac repressor. The lac repressor protein sits as such, blocking transcription. Notice there's now no inducible transcription. There's no making of messenger RNA. None of these proteins that metabolize lactose are going to be made. That makes sense because lactose is absent. There's no need to metabolize it. Okay, so that's a good thing. Let's say, by the way, this is what we call an example of negative control. This is a negative control of gene expression. You're stopping the gene from being transcribed. Let's say that all of a sudden lactose becomes available. There still may be some glucose there, but lactose is now going to be available. What's going to happen? When lactose becomes available, the cell is going to tend to want to start producing or expressing the genes that metabolize it. Okay, makes sense. If there's lactose there, why not do something with it? So when you start, what's going to happen is now lactose is going to be present. It can actually be metabolized into a form called allolactose. Just think of it as lactose. And it acts as what we call an inducer. Okay, it's an inducer. The inducer can now bind to the repressor. Okay, it can bind to the repressor, the active repressor. Let's visualize this. The lactose is going to now bind to the lac repressor. When lactose binds to that lac repressor, the lac repressor goes away. You've now removed the roadblock that was blocking RNA polymerase from coming in and transcribing these genes. So, in the presence of lactose, you now have expression of the genes. You've removed that block there, and you're now going to start translating and transcribing and translating these proteins that metabolize the lactose. That's the first half of the story. So basically, that lac repressor is like an on-off switch. It can flick it on or it can flick it off, the expression of the lac operon. This whole thing together is called the lac operon. The second half of the story involves glucose. So there's a term that's called catabolite repression, and that involves this system. What catabolite repression means is that in the presence of a particular catabolite, the cell's going to want to repress the expression of genes that would encode proteins that metabolize other metabolites. Here's the example. In the presence of glucose, the cell is going to want to repress the genes that would express proteins that metabolize lactose. This is how this works. Okay, so let's say the levels of glucose in the cell are going to drop. If the levels of glucose in the cell drop, it's going to effectively increase cyclic AMP concentrations. That actually holds true for both prokaryotic and eukaryotic cells. When glucose levels drop, there's signaling events that occur that jack up the cyclic AMP levels. Cyclic AMP is an important secondary messenger that we'll talk about a little bit more. Now moving to our figure, cyclic AMP is then going to bind to a protein called the cyclic AMP activator protein, or CAP. CAP. The binding of cyclic AMP to CAP is then going to activate this complex, this cyclic AMP CAP complex. It will then bind to the CAP site, and binding to the CAP site is going to activate transcription. So the CAP site is an activator site. You bind to this site, it's going to have positive control on gene expression. It's going to turn on the transcription and translation of the lac operon. So the way to think of this is like the dimmer switch. Okay, you have the off-on switch with lactose. If lactose is present, it binds the lac repressor, it leaves the situation, switch is on. We now have a dimmer switch where if glucose is absent, it's going to crank the dimmer up. When glucose is present, it cranks the dimmer down. So the switch is completely off when lactose is absent, so nothing's going to happen. If lactose is there, the switch can go on. But is the switch bright or is it dim? If glucose is present, the switch is going to be fairly dim. But if glucose is absent, through the cyclic AMP activator protein, it's going to turn the dimmer on to bright. So it's a wonderful way to regulate gene expression. It's the prototypical example that boards in the past have expected medical students to know. So I would give this a medium importance rating, probably low to medium importance, but it's important that you do take a look at this, particularly for the USMLE. Okay, in regulating gene expression, we discussed some events that occur. Now we're going to move from prokaryotes. We're going to jump over to eukaryotes. In regulating gene expression, there are other methods to do so. There were histone acetylases, and there were phosphorylation of histones. These types of things tended to obviously either activate or inactivate gene expression. The acetylation tend to favor gene expression, whereas the deacetylation, it favored more inactive or heterochromatin. There are proteins, we're going to talk about some, that change the folding, the DNA it's folding. It's going to put it together in a conformation that's either going to turn on gene expression or turn off gene expression. There are also DNA methylating enzymes. Remember, when DNA becomes methylated, it becomes inactive. So anytime you see DNA methylation, you should be thinking about inactive DNA or heterochromatin. Another distinction that needs to be made is that most genes, okay, in a diploid organism, most genes are going to be co-expressed. They're going to be co-dominant, meaning both your paternal and your maternal forms of the genes are going to be expressed off of both chromosomes. There are a couple of exceptions to this rule. Okay, the exception to this rule would be your X chromosomes. In a female, once again, you get the bar body formation. One of the X chromosomes, it becomes inactivated through lionization. That would be one exception to the codominant expression rule. The other exception are going to be in your B and T cells. Remember, in your immune cells, you get allelic exclusion, in which one, either your paternal or maternal copy of that gene encoding the T cell receptor or the B cell receptor becomes inactivated. So two major exceptions would be your B and T cells and in your X inactivation through bar body formation. Otherwise, expression of genes is going to happen from both your maternal and your paternal copies of particular genes. Now, in order to turn on transcription of a gene, there are sets of DNA sequence that are called enhancers. And just like the name suggests, an enhancer region, when bound by a transcription factor, is going to turn up the expression of a gene. So in eukaryotes, we have a particular set of sequences that are needed to turn on the gene. Enhancers often lie far away from the DNA sequence, the coding sequence itself. So as is depicted here, we have a promoter region for a particular gene, and then we have a region that's transcribed. It starts the +1 sequence. This contains promoter elements such as the CAT box and the TATA box. There's actually a UPE region, which is a GC-rich sequence we don't need to worry about. But notice that the enhancers are going to be far away, either far upstream or enhancers can lie downstream. So don't just assume that enhancers lie upstream for a gene sequence. They can also lie downstream as well. And this helps get, when protein factors come and bind to these enhancers, it folds the DNA just so that it can recruit the RNA polymerase and turn on transcription. That's the whole name of the game here, is that you're trying to turn on or turn off transcription. There are two types of transcription factors. There are general transcription factors that are general, meaning that every cell that expresses protein is going to be bound by these transcription factors. There's another set of transcription factors that are called specific transcription factors. It's the specific transcription factors binding to the enhancer regions that give specificity for gene expression, so that only when a specific protein is present, it could cause an enhancer, or I should say a particular environment is present or a particular condition is present, where a enhancer would bind to, a transcription factor would bind to an enhancer region, and then this would turn on expression of that particular gene. The general transcription factors are always going to be there, and they're going to be the same from cell to cell. Once again, it's the specific transcription factors that are different from cell to cell. Let's take a look at the actual picture of what's happening as transcription is being activated. You'll see in this particular figure that you have a DNA sequence. This DNA sequence has an enhancer region, it has a promoter region, it has a transcribed region. Notice that many complexes of proteins must come together just to turn one gene on. You have all of these proteins coming together. You've got the RNA polymerase, you've got transcription factors like TF2D, others that aren't even named. Okay, many of these specific transcription factors have to bind to enhancer regions. Many of the general transcription factors are going to bind to the promoter sequence. The whole idea is this complex forms, and then you get transcription at this transcribed region. Keep in mind that there's other sequences that are called not enhancers, but that are called silencers. Silencers can actually bind to the other regions of DNA, and as you would suspect, they prevent gene expression. So enhancer regions bind to proteins that turn on gene expression. Silencers, they're going to bind to sequences that turn off. They turn off gene expression. And lastly, just familiarize yourself here with the following terms, both trans factor and cis elements. You might read these in one of your questions. A trans factor is referring to a protein. A trans factor is a protein that can come, and in trans it can bind to DNA. The term cis element is referring to a DNA sequence that a trans factor can come and bind to. So cis element, trans factor, remember the elements, the DNA, the factor would be the protein. Here's a table giving some specific examples of transcription factors and then their requisite DNA response elements. Okay, we start out with the steroid receptors. We see that steroid receptors are usually proteins. They bind to a steroid. Okay, think of a steroid. What is a steroid? It's lipid soluble. It's lipid soluble, so it's greasy. It can just diffuse through the plasma membrane, and it can bind to an intracellular receptor. Having bound to that intracellular receptor, that complex can now go down into the nucleus, and it can bind to DNA. It binds to DNA at these sequences that we called hormone response elements, or HREs. This is going to turn on transcription. It's going to turn on transcription. Now, the protein class that these belong to is an important class. This has been tested, so make sure you're familiar. It's called a zinc finger protein. A zinc finger protein. Allow me to illustrate. This is an oversimplification, but perhaps we'll drive the point home for you. You could think of DNA as a double helical structure here. Kind of looks like a caduceus, so maybe you're familiar with this. A double helical structure to which there's going to be major and minor grooves that proteins can come in and bind to, the major and minor grooves. Zinc finger proteins are proteins that are coordinated with the mineral zinc. And this coordination with zinc gives them a structure that allows them to go fit in and bind to the DNA. Let me illustrate. The zinc finger protein is just going to go in and bind to the DNA. Okay, not exactly how it happens, but it drives the point home enough that zinc finger proteins are going to bind to the DNA, they're going to change the conformation of the DNA, and they're going to, in most cases, activate transcription. Another element is the cyclic AMP response element binding protein, or CREB, that binds to the cyclic AMP response element. We saw a little bit of this with the lac operon in prokaryotes. In eukaryotes, when the levels of cyclic AMP go up, it's going to tell the cell, I got to do something. What is it going to do? Well, it's going to change the metabolism of the cell. And it's going to do this through a couple of different ways. It'll do it through, number one, phosphorylation, or as we're talking about here, it's going to do it through regulating gene expression. So when the cyclic AMP response element binding protein is bound by cyclic AMP, this can go into the nucleus, bind to the DNA, and turn on expression of genes. This works through a different type of protein motif called a leucine zipper. You could think of leucines as being hydrophobic amino acids, and they're going to come together and form these hydrophobic interactions with one another. So this leucine zipper motif is going to be an important DNA binding motif. It goes around the DNA and links with itself, and it's like a little zipper, these being the teeth of the zipper, the leucine moieties. Another class would be the peroxisome proliferator activated receptors, or PPARs. These are going to bind to PPREs, or peroxisome proliferator response elements, receptor elements. These are highly important in lipid metabolism. Important for lipid metabolism. There's some diseases here, such as Zellweger's disease. Okay, not Renelle Zellweger, but Zellweger's disease. It's when there's a problem in degrading your long-chain fatty acids within peroxisomes. That's what goes on there, is long-chain fatty acids become degraded. And if there's a problem with that, they build up and it causes Zellweger's disease. Another important point about these is that they're the target of your fibrates. Okay, people with altered lipid profiles oftentimes will be placed on fibrate or fibrate therapy. Things like gemfibrozil will be prescribed. This is going to interact with this system, which you'll learn about in pharmacology, but it will interact with this system to help alter the lipid profile in a more positive manner. Finally, homeodomain proteins. Homeodomain proteins are a little bit different. I'll illustrate. They primarily regulate gene expression during development. They work through a different type of motif called a helix-turn helix. So there's a DNA double helix, there's a little turn, and then there's another helix. So something with a conformation like that is going to be able to slip right onto the DNA and start changing its shape, altering gene expression. Let's look at a specific syndrome called Klein-Vardenberg syndrome. In Klein-Vardenberg syndrome, there's a defect in a gene called PAX, called PAX. A PAX gene is a type of gene that we call a homeobox gene. Not a homeboy gene, a homeobox gene. A homeobox gene. A homeobox gene encodes a homeodomain protein. What's important about these homeodomain proteins? These homeodomain proteins are themselves transcriptional activators. So think about during development, during embryological development. You've got all these pathways being turned on simultaneously, and all this coordination that has to happen to get the limbs to grow properly, to get the fingers popping out in the right spots and whatnot. All this regulation. Well, this is the job primarily of homeobox genes. They're going to encode a homeodomain protein, and the homeodomain protein, I can show you, is going to bind to enhancer regions. When the homeodomain protein binds to an enhancer region, that's going to turn on transcription of the genes. So one homeodomain protein can bind to many different, the enhancer region for many different genes. In this way, you get amplification of a signal. What are some of the symptoms of Klein-Vardenberg syndrome? As I said, a defect in the PAX gene. You would see the dystopia canthorum in the eye. You would see the pigmentary abnormalities. You see that in the hair and in your skin. You could get congenital deafness. And by and large, a major presentation is limb abnormalities. So all of these problems, developmental problems, because you couldn't regulate development through a homeodomain gene. Another example of how gene expression is regulated in humans. This is the one I'm showing you now is probably the most common way in which most genes are regulated. And it involves, once again, these zinc finger proteins. Remember that term. Zinc finger proteins are transcriptional activators. Most steroid hormones, your thyroid hormones, your vitamins A, vitamin D, which can activate transcription, these all function through this zinc finger motif to activate transcription in the nucleus. We'll look at two examples here. We're going to look at cortisol and we're going to look at glucagon. Cortisol, a steroid hormone, is lipid soluble. It very nicely can slip through it's a really greasy character here. It can just slip right through the plasma membrane, in this case an hepatocyte. It will bind to an intracellular receptor. This cortisol receptor complex now can act as a zinc finger protein, which goes to the nucleus and turns on the growth response element, or I should say binds to the growth response element portion of DNA, which will turn on transcription of many different genes. One particular gene here is the PEP carboxykinase, all right, phosphoenolpyruvate carboxykinase gene. You know that gene as an important enzyme involved in gluconeogenesis, or the making of sugar. Okay, it happens almost exclusively in hepatocytes, gluconeogenesis. What causes it to happen? Stressful conditions cause it to happen. You're in a stressful condition. Your adrenal gland starts releasing cortisol. The cortisol is going to now signal to your liver to start pumping out glucose. Right? Your brain needs that glucose. Your red blood cells need that glucose. So this is going to be regulated here at the transcriptional level through the cortisol binding to a response element. So if more PEP carboxykinase gene is being made, you have more messenger RNA present. PEP carboxykinase, as we'll talk about in gluconeogenesis, its job is to take oxaloacetate and convert it into phosphoenolpyruvate, or PEP. PEP carboxykinase, one of the important steps in gluconeogenesis. The rest of the steps of gluconeogenesis cannot be undergone, and finally glucose can be released from the liver cell. So one of the key ways the liver starts pumping out glucose in response to a stressful situation. Okay, so low levels of glucose going to signal cortisol to undergo this pathway. The enhancer is bound. Okay, therefore you're going to start increasing the levels of glucose that are being formed. The second way that hormones can regulate these types of pathways would be something like glucagon. That's shown over here. Glucagon is a water-soluble hormone. It can't diffuse through the plasma membrane. It has to give a little knock on the door here on the cell. It has to give a little knock on the door on the receptor and cause something to happen. Give it a little tickle. It gives the cell a little tickle. This is going to function through something called a G protein. A G protein we'll talk about. But the net result, of course, is going to be increasing in cyclic AMP levels, cyclic adenosine monophosphate. Okay, cyclic AMP is just the AMP nucleotide that's formed an internal phosphodiester bond and now acts as a signaling molecule. Cyclic AMP is going to activate protein kinase A, which then can phosphorylate a protein called cyclic AMP receptor element binding protein, or CREB. Cyclic AMP receptor element binding protein. We discussed this on the last slide. When phosphorylated, CREB can then go and bind to cyclic AMP response elements, which lie upstream of a number of different genes. These are enhancers that occur not just on PEP carboxykinase, but on all sorts of genes that are important in regulating gluconeogenesis. So, by binding to these response elements, once again, you get activation of transcription of genes, which is going to then upregulate the amount of glucose that can be made and then secreted from the hepatocyte. There's an important enzyme here we'll discuss in the next section. It's called adenylate cyclase. Adenylate cyclase is the key enzyme, once again, that's going to turn on cyclic AMP production. So in summary, the controlling of gene expression occurs at many different points along the gene expression pathway. Remember, gene expression involves both transcription and it involves translation. You can actually even involve the DNA level. So just by taking the DNA and packing it really closely together via methylation of DNA or affecting acetylation or affecting other ways, you can just pack the DNA really tightly and prevent it from being available for the polymerase to come in. Gene amplification. Your cells can actually amplify the number of genes that are present within the cell, and this is one way to start goosing up the number of proteins that are being made. Some tumors, this happens. Some tumors, during replication, the number of genes begins to increase, and therefore you get extra protein production, and that can be a problem. Once again, transcription factors, as we've discussed, mRNA processing can be affected. You can affect the way in which RNA is being processed through alternative splicing, so that instead of, for example, membrane-bound antibodies being produced by the B cells, you now get secretory antibodies being produced. The rate of translation can be affected as well through mechanisms that we don't need to discuss. They won't test you about. Protein modification can be made. Many different ways that happens. I think we talked about collagen, important modifications of collagen that have to occur. And finally, the degradation rate. The rate at which proteins are being made is regulated, but also the rate at which they're being broken down is regulated. Not as much known about the degradation pathway, so I wouldn't worry about that as much. This concludes Chapter 5, talking about the regulation of gene expression.